Okay, so what you can see is we have the Punnett score on the board. So we're going to do a quick test. This is called PTC paper. And the PTC paper is one that checks to see what kind of genetics you have. So we're going to do a quick test to see if we are a taster or not. And that's on the next slide here. And so what we're going to do is, we talked last time about how we had eye color, how we could have like a dominant color and a recessive color, right? Remember that? Which one was dominant?
One you can see.
Brown, the one you can see. Yeah, what do we call that when you can see it?
Phenotype.
Phenotype, excellent, phenotype. And we call it what when it's hidden in our genes?
Genotype.
Genotype, excellent. So we're going to do a quick taste test. So right now we have paper in front of us that'll tell us what kind of taster we are. We have 3 gene types. If you're a capital T taster, that means you're a dominant taster. And what that means is that when you put this paper on your mouth, you will get a bitter flavor. That's what I've been told. I personally am double recessive, so I can't taste it at all. So when I first did this genetics test in one of my classes, what my lab partner and I were kind of like, okay, what is this going to taste like? She's like, you go first. And I put it in my mouth and I go, oh, it's a trick. It's just paper. There's nothing on it. And then she put it in her mouth and it was super bitter because she was a taster. So if you taste a super, super bitter flavor, like just touch it to your tongue, see if it tastes bitter, that means that you are a double homozygous, meaning you're 2 dominants. If you taste a little flavor, then you're what we call heterozygous, meaning a dominant and a recessive. If you don't taste anything, you're recessive. You guys ready to try it?
Yeah.
Yeah? Okay, so take your mask down really fast and go and touch it to your tongue and see what kind of flavor you get.
Now?
Yeah, go ahead. And you can throw them in the trash when you're done. It's just like that. I see some faces of tasters. Anybody a non-taster? Who in here is a non-taster? Yeah, okay, so what this means is if you're a capital T, that means you're a taster. So that's what's on the board right now. Phenyl carbamide tasting. It's a taster. And then if you're a lowercase t, you're a non-taster. So what this means is your taste buds do not detect that enzyme that's on the board, or you don't produce an enzyme to break that protein that's on the board, I should say. So your body doesn't taste it. This is why people have different tastes. Some people like things super hot. Some people like things spicy. Some like them bitter. A lot of times what we see is that people who are recessive, we tend to like things that are like a little bit spicier or more flavorful because we don't get as strong of a flavor. People who are double tasters, they tend to like things kind of in the middle range. And then people who are this mixture, they can go either way. They can go, super taster or non-taster. Do you like cilantro?
Yeah.
You do? Okay. A lot of times if you're a super taster, you don't like cilantro, which is interesting because it's almost too strong. It tastes like soap to some people. Have you guys ever heard that? Cilantro tastes like soap?
Yeah.
That means that they're probably a homozygous dominant. Yeah.
You like cilantro.
Exactly. I love cilantro. Yeah. Put it on like a truckload. I love it. Yeah. So and this is why some people are able to do things like this, like when you go wine tasting, some people can taste all these flavors, and then other people, like I'm one of those people, I'm recessive, I don't taste anything. I'm like, I don't get anything, So it's kind of explains some of our genes. Isn't that cool? Okay, so this is going to basically be then what we see when we do any kind of genetic testing or phenotype testing. We're looking to see what kind of genes you carry. So let me ask you this. Let's go back to a summary. What does it mean when I tell you that you are carrying a gene? Is that the sum total of all of your genetics? Is that a segment of the chromosome coding for a product, a segment of the nucleus, or the study of heredity? B. Yeah, it should be B, a segment of the chromosome coding for a product. True or false, viral genomes can be based on DNA or RNA. What do you think? If I'm a virus, can I have DNA or RNA?
Yes.
Yes, exactly. So viral genomes can be either DNA or RNA. Now, genomes can vary a lot in size. The smallest virus out there has a genome that is only about 4 to 5 genes long. That is tiny. That is such a small genome. But let's think about a virus. A virus doesn't need to carry a lot of information because once it gets inside of our cells, it's going to use our cells to do all of its work. So it doesn't need to have information about how to, manufacture things that it's going to steal from its host. It only needs to have information about how to make the host cell do what it wants. That's really all it needs. Some bacteria, as I mentioned before, have small genomes. E. coli has about an average size, about 4,000 genes. If I stretched out the genome of E. coli, it would be about 1 millimeter. That's about the thickness of a credit card. You look at your credit card or driver's license, that thickness, that's the entire genome of E. coli. Isn't that cool? I'm looking at it from the side, so you can get an idea of how small it is. Our human cells, with the size of our genome, if we stretched it out, it would be about 6 feet. So today, I have high heels on, I'm probably about 5'6", 5'7". So imagine like 6 feet. That would be my entire genome stretched out, which would be pretty cool. So this brings us to something that we refer to as the central dogma. And again, if you printed out your slides ahead of time, there's a lot of stuff in between you can cross off, just skip to this one. The central dogma is this idea that DNA is going to dictate what proteins are being made in our cells, what amino acids and proteins are being made in our cells. So in other words, the DNA is the one who drives everything. It's the one that's going to be the master, as we talked about before. It's going to be the one that tells what to do. And then from the DNA, we're going to make RNA, and that RNA is going to go to which organelle in our bodies? Which organelle does the RNA go to make proteins?
Ribosome.
Ribosome, excellent. And remember, the bacteria also use ribosomes, but they're not organelles because they're not membrane bound. So that's the difference again between those organelles. And we're going to make our protein that's present. So all organisms are going to use the same basic process to make or express the genes that they have. We are all an accumulation of our genes. Our genes say what color our skin should be. Our genes say what color our hair should be. Our genes say what color our eyes should be. It is all protein based. That's all it is. It's just a protein. And so our flow is always DNA to RNA to protein. This again is called the central dogma because every life form out there follows this pattern. If I'm a virus and I have RNA, I'm going to go from RNA to a protein. Okay? If I'm a human, I'm going to go from DNA to RNA to a protein. All right, so I'm always going to do this flow no matter what organism I am. This central dogma then is part of the reason why vaccines like Pfizer and Moderna are getting approved. So a lot of controversy, of course, is around anything that involves RNA. It seems like those vaccines kind of scare people. And one of the things I've read or heard a lot is that it will change our DNA. But that's really impossible because of the central dogma. The central dogma says I go from DNA to what? RNA, and then to a protein. So if I inject a vaccine that has RNA, is it suddenly going to go backwards? No, that's against biology. We can't go backwards. There is only one type of virus that we know of that can go backwards, and it has to have a whole bunch of special enzymes to do this process, and we will never turn that into a vaccine because you're right, that would be possibly risky with those enzymes that are in place. We'll talk about that virus. So the idea behind this then is we get a one-way street. We can't suddenly turn around. It's like if you ever go to San Luis with all their one-way streets and suddenly start driving the wrong way. Don't do that. Okay, don't do that. They get really grumpy if you do that. So, yeah.
How would the Johnson & Johnson?
Good question. So Johnson & Johnson is older technology. So Johnson & Johnson, what they do is this is what they do for a lot of vaccines. They use what's called a vector. So they take a cold virus, they pull out all of the genes out of it, and they insert that one segment of protein that they want to make. And then what we're making is the spike protein of COVID, the little spikes that are on it. And then what they do is they insert that into the vaccine. So when you get an injection with Johnson & Johnson, you're getting the actual protein. The only difference with Moderna you're getting and Pfizer is you're getting the mRNA, which will then go to your ribosomes and make the protein. So what's the point? Why do we want a protein? Well, here's the cool thing. When your body makes that protein, it kind of looks at it and goes, wait a minute, this isn't one of my proteins. What is this? And your immune system gets called over and it says, wait a minute, that's not one of ours. And your immune system destroys the proteins that were made because they realize it's not one of ours. And this is where it gets good. Now they're going to make what we call memory cells that are going to recognize those proteins in the future. So if you get exposed to COVID again, your immune system goes, I recognize that. That's a protein structure we didn't like. And it attacks it as soon as you get exposed, instead of waiting and trying to figure out what's going on while the virus is sneakily replicating. Does that make sense? So it's kind of like giving you, I always tell people, think of vaccines as like a most wanted poster. What is a most wanted poster? You may remember those at the post office if you ever go there anymore. They have pictures of people that, like the FBI are looking for. That's kind of what a vaccine is. It shows your body, look for this. If you see this, get rid of it. And your body's like, okay, I'll do that. And that's what it does. So, yes.
So, I have a question.
Yeah.
Since Pfizer and Moderna, they're inserting like the RNA to make the proteins, is that why it's more effective than like the Johnson & Johnson because you're just putting in the proteins?
Great question. So what we suspect, this is the idea behind it, is we suspect that Pfizer and Moderna, because we're making the proteins, are going to give a longer, more memory cells, a longer immunity. That's what we're thinking. Johnson & Johnson, we think, will have a shorter term immunity. Our body makes a certain amount of protective cells based on how much protein they're exposed to. So if we're making that protein, we have a chance of being exposed to a higher amount than if we're just injecting it. Does that make sense? Yeah, so that was the thought, the logic behind it. Also, it takes a lot of work to use a vector. You got to remove everything from that vector. Like I said, it's a cold virus. We got to move everything out of there to make sure we don't get you a cold. We don't want to do that. So we got to get rid of all the genes that are in there and then insert what we want and keep it stable. That takes a lot more work, which is why Johnson & Johnson was a little slower to the market. All right. Any other questions? This is good. Can you still get COVID if you're vaccinated?
Yes.
Yeah. What is up with that? Okay, well, let's talk about that. If I'm vaccinated and my body is ready to fight that protein, but I get exposed to somebody with a high viral load, meaning like I get 1 million particles hitting me at once. Okay, this is the difference between walking into your house and picking up 1 Lego on the floor versus like 1 million Legos on the floor. Are you going to be able to pick up 1 million Legos? Yes, but it'll take time, right? It's going to take you some time. This is why if people are exposed to a high viral load, they will get a little bit, they'll feel a little run down, they'll feel a little sick because their body's picking up, getting rid of all the viruses they were exposed to. The beauty of it, though, is that you recover faster because you already have that immunity kind of in place. All right? So hopefully this kind of explains some of what is going on out there. There's a lot of confusion about how these vaccines work because of that central dogma flow. If they're not understanding it's a one-way street, it's easy to get confused. And it's easy to get scared. You start thinking that maybe they're changing their DNA. The other one I heard was that if they're putting stuff in your body that's RNA, it can be a tracking device. You guys heard that one too? I'm like, you're already being tracked on your cell phone. I hate to tell you. No, I mean, if you want to panic about tracking, that's the easiest way. They can ping your phone anywhere. So it's not something you have to worry about. But I'm kind of wanting to make sure that we kind of get that understood. One thing I will tell you, I will never tell you you should do something or shouldn't do something. I don't feel like I ever need to do that. You guys are adults. You can make your own decisions. My job is to offer education and to say, here are the facts, and then that way you can make an informed decision as opposed to, reading Jenny McCarthy or some artist or actress who's saying, well, this is what I'm doing and you should do the same. Everybody has to make their own choices. All right? So the central dogma then. The central dogma is going to be this gene expression. And so it's based upon using information from the DNA to make a protein. And again, the DNA is the one who runs this whole show. So the DNA is going to be directing everything. As I said with vaccines, we can jump in the middle, but I can never go back to the DNA. Especially in our cells, we have it in the nucleus. It's not like it could ever go back into the nucleus like that. Now, the transcription process then is going to occur, and as you can tell, I cut a lot of slides out on this part. It's going to occur where we're going to make an RNA molecule based on the DNA template that we have. So in other words, I have my DNA. My RNA goes in and reads the DNA, makes a copy of the information that's there. And then what's going to happen is that RNA copy is going to leave the nucleus for us. For prokaryotes, it's going to leave the nucleolus, and it's going to move out to the ribosome. And now in the ribosome, that RNA can get read and processed. So that's what we're looking for. That process of the RNA reading the DNA is referred to as transcription. Think of it as like you're transcribing notes. It's transcribing the messages that the DNA wants. Okay? Now what's going to happen is the RNA is going to attach to our DNA, build a copy until it reaches a stopping point. That stopping point is a set of codes in the DNA that basically says stop here. The way this works is kind of like when we were kids, when we read fairy tales. There's a part on the DNA that says once upon a time. Remember how fairy tales used to start with once upon a time? Okay, that tells the RNA attach here and start here. And the RNA will read the DNA until it hits the section of DNA that says the end. And then it'll fall off, and it's now ready to leave and move on to the ribosome. Now, we refer to that section of DNA that says the end as the terminator sequence. But we don't want to say it boring. How do we want to say it? We want to say it like Arnold Schwarzenegger. We want to say the terminator. It's the terminator sequence. Yeah, isn't that fun? Right? I mean, this is pretty fun stuff. Okay, so then that RNA drops off, and it moves to the ribosome. So the way that the RNA is going to get processed in the ribosome is it's going to get read 3 bases at a time. We refer to 3 bases at a time as a codon. So 3 bases at a time is a codon. And so what's going to happen is my DNA is going to have bases. I'm going to see if I can change my background here for my Zoomer so you can hopefully see on the board. We'll see. Try to do something here. Okay, hopefully. I'll try to write big. All right. So if I have DNA, let's start with something like AUG. Okay, AUG is my DNA. Let me see if I can do a darker pen. Sorry, I'm just trying to see if I can find something that works for everybody. All right, let's do this. AUG. Okay, so that's my DNA code. Okay. Now I'm going to read this and convert it into RNA. Well, remember our rules about RNA. Remember the bases they have. If I have an A here, what's my RNA base going to be? Does RNA have T?
No, it doesn't.
What's that?
Want to be ATG for DNA.
Oh, yeah, you're right. You're right. ATG. You're right. Sorry. Thank you. You caught my error. Thank you. We'll say ATG. We'll say, actually, let's do ATC. We'll say ATC. Okay, ATC. So A is going to be U, right? That's going to be my RNA. Thank you for catching that. That's going to be my RNA. What about the T? What base does RNA have that can read a T? A. How about the C? G. Excellent. So this is my mRNA, okay? So this is going to be the codon that will move to the ribosome, okay? And there in the ribosome, it will be processed. All right. Now, once it gets to the ribosome, we're going to have another type of RNA read it. The type of RNA that's going to read this RNA is one referred to as tRNA. This is the one that comes in and out of the ribosome trying to bring in a match to whatever sequence is presented to it with the mRNA. Okay? So if I'm tRNA, what would I bring in if there is a U? What binds with U in RNA? A, excellent. What binds with A? U. I hear whispers. What binds with G? C. Okay. So my tRNA, my anticodon. Now, why is it called an anticodon? That sounds like it's against it. All that means is it's the opposite. Okay. It's the matching pair that goes to this. All right. That tRNA, when it zooms in, it's going to have the codon that matches. And guess what's attached to the tRNA? Well, tRNA, when it comes in, brings a friend. What's attached to it is an amino acid. Ooh. What do we use amino acids for? What do amino acids build up to create? Proteins. So this is how this is going to work. So I'm going to have mRNA come into the ribosome. I'm going to have tRNA match up to it a codon at a time. How many bases are in a codon? 3. Good. So 3 bases at a time. Each tRNA that can match those 3 bases comes in with an amino acid. And I'm going to have these amino acids linked together, and I'm going to make a protein. Okay, we're going to look at this process. Does everybody get where we're going, though? We'll make sure we got our game plan. Okay, yeah. So here's a picture of DNA to mRNA to an amino acid. This is a chart showing the mRNA bases. Now, the reason I point that out, sometimes on the internet, you'll pull up a chart and you'll be like, this is not the chart that Professor Hadley showed me. It's because it's showing you the tRNA. It's showing you the anticodon. So you have to see if it's the mRNA or the tRNA. Okay? It'll just affect things a little. It'll be different bases. Okay, so let's look at this. When we first look at this chart, it looks terrifying. Doesn't that look scary? And you do not have to memorize this. Okay, if I ever asked you to code, I would give you a chart like this. All right? Or I would give you whatever charts you needed. Let's look at how to read it, though. It's actually not that hard once you kind of see the pattern that's present. Okay, the first thing is that it's arranged in blocks. So notice there's a block here that has a U on it. I'll do this for my homers. U here. And then there's a block here that has a U on it. And then there's a block at the end that has other letters way over here. This is telling you what the sequence is going to be. So, first letter is U. So everything in this row is going to start with a U. See how that works? Second letter at the top, this one's U, so all the middle letters should be U. This one's C, so all the middle letters going down should be C, A, and so on. Make sense? And then on this end, this is showing what the last letter will be. So here's a U, here's a C, here's an A, here's a G. All right, so just showing you, so in case you have to look a sequence up, that's how you can quickly find it. You would look at the first letter, that's all of these first letters, C's. The second letter, that's all the middle ones, and then the third letter. All right, so there's a couple of things that we should notice in this table. First of all, this amino acid, AUG. AUG is a very special amino acid because it is methionine. Methionine also is our start codon. A start codon means this is the one from DNA that says once upon a time, in the beginning. Methionine is our start. Okay? There's a couple others in here that we should look at. Do you see these 3 up here? What do they say? Anybody see what that says? Stop. Yeah, so these 3 over here say stop. The stop codons are the ones that are the terminators. They're the ones that are the end. What that means is if I have a stop codon, there is no matching tRNA for it. So when I hit this point, I know that I'm done transcribing because I don't have a match. I can't match anything to it. Okay, it's like having one shoe at home. Can you wear that one shoe? Well, no, not unless you have its pair, right? I mean, you can walk around with one shoe on, but your kids are going to think you're crazy, right? Or your roommate or whatever. So it's the same idea where if I don't have a match, I can't progress. Okay, so that's what we're looking at here. So let's put this all together. Okay, so we have those codons that are going to be going into the ribosome. And here's our ribosomes. This process that we're going to be talking about moving forward is the same whether I'm a bacteria, a prokaryote, or a eukaryote. This is the central dogma. It's all life. Okay? What's going to happen is my ribosomes, as we mentioned before, exist as a large subunit and a small subunit. And what they will do is when RNA comes up to them from the nucleus, so it would be the mRNA, they're going to close down and bind together on top of that mRNA. It's kind of like putting a hamburger bun together, like sandwiches, the mRNA. So the mRNA is like the patty in a hamburger, okay? And now what we're going to do is we're going to read that mRNA one codon at a time. Each codon will be matched up to a tRNA sequence, okay? All right, so in our ribosome, we're going to have 3 sites. We have A, P, and E. Thankfully, it's an acronym that's easy to remember. APE. Isn't that easy? I can remember that, right? Think about this. Learning this stuff is monkey business, right? It's like crazy. Bananas. So we can remember apes, all right? The A site is going to be where our mRNA is going to first arrive. When you see A, think of arrival. It arrives here, okay? The P site is where I'm going to build my protein. So when you see P, think of protein. So the P site is where I'm going to build my protein. And the E site is where I'm going to exit. So I'm going to leave the ribosome. And we're going to see this process in action because it's pretty cool to watch. So what I'm going to do now is I'm going to have finished my transcription. Remember, that's where RNA reads the DNA. And I'm going to move to the ribosome and I'm going to start my next process, which is translation. Translation. So on translation, what this means is I'm going to be matching tRNAs up to my mRNA, one codon at a time. So my mRNA is going to arrive in the A site. A tRNA is going to match up to the codon that's there. And then they're going to move together to the P site. And I will show you a picture of this. The amino acid that comes in with the tRNA gets left in the P site. It builds the protein there. And then we're going to exit and leave the ribosome. So let's look at this in pictures. All right, so first, here was my first codon. Do you see how it's a start codon? AUG? It's sitting here in the middle. Okay, so when it first arrived, it arrived here in the A site. And what happened was we matched the matching tRNA, the anticodon, to it. You see how that works? So if I'm AUG, up here I will be UAC. So I'm looking in that middle site, right? Now, check it out. There's the amino acid attached to it. The amino acid is methionine, right? So in this middle portion, that's the methionine. Everybody see that? Okay. So as this is being processed in the P site, my next codon is being read in the A site. I have the next anticodon swooping in. It's going to match and bind to it, and then they're going to move over. This one will move towards the exit. This one will move to the P site. You see how it's kind of like a little conveyor belt. Okay. All right. So here we go. We have methionine still sitting in the P site. We have leucine now in the A site sitting here. All you need to know is that I have an mRNA and a tRNA together. These 2 amino acids are going to see each other and they're going to bind together, and I'm going to start building my protein chain. Here's what it's going to look like. In the P site, I now have both of the amino acids that I brought in. The amino acids stay behind in the P site even when the tRNA that brought them there moves on. It's kind of like dropping somebody off. You know, you drive somebody, you drop them off, and then you leave, and they stay behind. All right?
So the tRNAs leaves?
The tRNA is going to leave with that mRNA. They're attached together now. And so they're going to move together to the next site. But the amino acid that came in with them stays in the P site. Okay? And if this is confusing, ask now. Because this is kind of like metabolism. The first time you see it, it's confusing. And then you start to feel better about it. Okay? So it's okay. Okay. So now what's going to happen is my next amino acid processing is going to start in the A site. So here I have my next codon, my next tRNA with the anticodon, and attached to it an amino acid. And we're going to keep doing this process, bringing things through. All right, my tRNA is going to leave the exit site over here. I still have my protein building in the P site, and my codons are pulling through like a conveyor belt. All right, let's keep going. Check out my P site. Do you see how I'm building a chain of proteins? This process is going to keep happening for each codon. So we're just building a chain here. So as I keep processing, this mRNA is going to keep moving through until I hit a stop sequence, a terminator. All right. So I'm going to keep pulling this through. Now, this is why I was saying some proteins can be really long. We can have hundreds of codons that we're going to be processing. So this process could take some time while we're pulling this through. It seems like it would be really clunky and slow, but this happens in a fraction of a second. Matching up a tRNA to an RNA occurs like that. Our bodies are built to do this. So it's actually a very streamlined process. What's helping them find their partners are little enzymes behind the scene that are matching things up. All right? So our last stage is the termination stage, and that's, again, where we're going to hit a stop codon. So a stop codon is where I told you before we don't have a match to it. It's like having one shoe. So my mRNA comes in, and I don't have a tRNA to match up to it. So that's going to tell the ribosome that we've reached a stop, and my ribosome is going to separate at that point. It's like taking the hamburger buns apart. When that happens, the protein that was in the P site that I've been building gets released. All right, so it's like taking my hamburger apart. All right, so here we go. Here's my stop codon coming in, UGA. There are no matching tRNAs for it. Nothing can match up to it. So this is going to tell the ribosome we're done. So the ribosome is going to separate. So here's the ribosome, my two hamburger patties separating. The mRNA that's here is going to get released, this chain of mRNAs, and it's going to get sent to the recycling center. Which organelle is that in us? Lysosome. Excellent. So it's going to get sent to our lysosome because we're done reading it. In bacteria, what they do is they kind of grab it, fold it up, and they spit it out of their cell. They're like little litter bugs. They spit things out all the time, so they just spit it out of the cell. And then, as our ribosome separates, look what gets released. Our brand new protein with all of the little amino acids that have been put together. All right? Does that process make sense then? Okay, let's make sure we can put it together. I go from DNA, and what do I make from my DNA? mRNA. Excellent. mRNA. Where does my mRNA go? Into the ribosome. Good. Is this the same for prokaryotes and eukaryotes? Yes. This is the same for all life forms. Okay? All right. So now I'm in the ribosome. Where does my mRNA arrive? What's the first site that it arrives at? The A site. Excellent. And while I'm in the A site, I'm going to get a matching what? Codon. And who's bringing in the matching codons? The tRNA. tRNA is bringing in the matching codons. What's attached to the tRNA? Not a code. What do we call that little piece that's attached? Amino acid. Good. An amino acid. And when I put a whole bunch of amino acids together, what do I build? A protein. That's my goal here. Excellent. Okay, so I match in the A site. I move to the P site. When you hear P, what do you think of? P for protein. I'm building proteins here. This is where I'm going to leave all the amino acids. They're going to stay in the P site. And then where does my mRNA and tRNA move next after the P site? To the E site, and then they exit. When does the process stop? When it hits a stop codon. When it hits a terminata. You have to say it with the Arnold Schwarzenegger voice. It just makes it so much better. Okay, when I hit a terminata, I stop. All right? Why do I know, or how do I know I've hit a terminator? What's my sign? There's no tRNA match. It's like having a shoe without a match, or a sock without a match. We all have those at home, right? We just eventually give up and throw them away, right? Or else you wear mismatched socks, whatever. Okay. Does that help to kind of go through that? Okay. And hopefully everybody at home is doing good too on the Zooms. Okay. So, can things go wrong? And the answer here is yes. And this is where we're going to skip a lot of slides if you print it out ahead of time, because I used to have more information about this stuff, and we just don't need it. I've streamlined it. So, where can things go wrong? Things can go wrong when we have a change in our genetic material. So, in other words, if I have a change in my DNA itself, in my sequence of DNA, that means that my mRNA is going to be changed, my tRNA is going to be changed, and my ultimate protein is going to be changed. Okay? How can I get a change in my DNA? Well, usually it's from exposure to what we call mutagens. Something that can cause a mutation in the DNA. This is what that Prop 65 warning is. Have you guys ever seen Prop 65? Yeah, it's everywhere, right? No matter what you buy, it has a Prop 65 warning on it. Even car seats have Prop 65s on them. What is Prop 65? It's telling you that some component in the manufacturing of that process, whatever they made, maybe a car seat or whatever, had a potential mutagen in it. Now again, it depends on what it is as to what the risk is. Some mutagens have to be ingested, some have to be inhaled, some have to absorb through the skin. It's going to depend on that process as to what mutagen they are, okay? So in this case, a mutagen can be anything that causes a change. We also can have spontaneous mutations. Spontaneous mutations are where, for some reason, the DNA just will have a base change. It'll have a mistake that'll occur. A lot of times we see that with aging cells, with mitosis, something wasn't copied right. And so we get an error, just like we're not all perfect when we copy stuff, we might have an error there. So there's different kinds of mutations out there. There's missense or point mutations. This is where I get a change in one base. And so what that means is I'm going to get a change in my codon, which means I'm going to get a change in my amino acid. Now, I did add a little information to this slide. If you printed this out ahead of time, pull your phone out and you can snap a shot of this slide. That might be a little bit easier because it does have the examples, because I thought the examples were helpful. So, in this case, maybe I want the sentence to read, "The cat ate the rat." That would be the correct DNA. And notice I chose 3-letter words for codons. Everybody appreciate that? It took a little time. Okay. The cat ate the rat. Then we come down here. Here's an example of a point mutation. I've changed the E for a Y. Now I'm speaking Old English. Thy cat has eaten thy rat. You know, it's like whatever. Yeah, we switched to Old English. Okay, so my body or my cells are going to process this, and we're going to have an error here. One amino acid will be different. It's coding differently. Okay? Down here, the cat ate the bat. Whoa, now I've changed the sentence completely, right? But I'm still just changing one amino acid. Okay? So what effect will this have? It really depends on what the amino acid is that was changed. When I start changing things in my proteins, I can affect the way they fold. I can affect the way they work. This is where we start talking about genetic diseases, where we talk about things not forming right in the body. Okay? Sometimes a tiny point mutation won't have any effect. It's so little, the rest of the protein will be okay, and we can kind of hide it. Other times, it can be huge. Where we see a huge change is with the nonsense mutations. And again, if you have your phone out, snap a picture of this one if you have the old notes. Nonsense mutations are where we get a base change at first, but unfortunately, this base change results in a stop codon. It makes a terminator. So now what happens is that maybe my sentence was "the cat ate the rat." I had my point mutation from earlier, so I have "thy cat ate." And then I do a switch here, and this becomes a stop codon, and I don't even get the rest of the sentence. My stop codon that hit here, and you can see here it's the yellow A, everything that comes after it gets stopped. Do you see where this would have a bigger impact? So maybe my protein was supposed to be 400 bases long, and I only got to 200, and I hit a stop codon. Ooh, that is bad. So the protein that I'm going to end up with is completely different now. Okay? Now, sometimes these mutations, again, they might not be huge. Maybe they're the mutation for red hair, right? Maybe they're the mutation for multicolor eyes. Have you ever seen that where somebody has a a green eye and a blue eye, or a brown eye and a blue eye? So sometimes these mutations can just be a little bit different, okay? But again, sometimes they can be pretty scary. Here's another one that we don't want to have happen. This is called a frame shift mutation. A frame shift mutation is where we're either putting in an extra base or we're removing a base. So something wasn't copied right. Now, if I do that, it's going to shift all of my sentences from here on out. So, for example, here's my sentence, the cat ate the rat. Now, I've inserted a letter B, and I know that's not a code that's in DNA. I know that B is not in DNA. It's an example, okay? So I've inserted an extra letter, an extra base here. Everything shifts. So here's CAT, but notice it got shifted, so it's no longer together. You see that? So here's the B that was inserted. Here's CAT, but it got shifted down. So now, from here on out, every single codon is going to be a different one. It's going to be incorrect. All right? So again, these can have a big impact on what happens with our cells. So mutations can be quite dangerous. In our bodies, when we have DNA replicating, and in bacteria when they're replicating DNA, we get a spontaneous mutation rate of about 1 in a billion. Now, again, some mutations are good, and some mutations are harmless. We don't even know they're there, and some mutations are bad. Some mutations give us immunity. This is why some people were immune to the Black Plague when it was circulating in Europe. They carried a different gene. They had a different mutation type that made the plague hard to infect them. This is why some people were naturally resistant to COVID, right? So that's a newer one. Some people have different genes than others. This is why some people, unfortunately, have passed away from COVID. Their genes weren't able to protect them. So their immune system wasn't able to react in time. So all of these mutations and things can have an effect on the life that we live. Now, how do we get these mutations? As I said, they can be spontaneous, or we can get exposed to a mutagen. An example of a mutagen would be something like ionizing radiation. This is something like an X-ray, a UV, a gamma ray. We know from experience that whenever we're exposed to these, we can get a slight mutation in the DNA depending on the exposure. Now, again, sometimes these exposures are ones that will result in nothing serious, and then other times they could potentially be very dangerous. This is the case that occurred in the 50s. There was a doctor named Alice Stewart who was working, she was an epidemiologist who was studying the impact of diseases in health. And she started doing an interesting study. She found that a lot of people in cities were showing an increasing number of birth defects with their babies. And she started trying to figure out if there was a link. Was it the water? Was it the food? What was it that was causing this increased number? And she started doing surveys at different hospitals. And what she found was that hospitals that were more affluent had higher mutation rates, higher birth defects. She started trying to put this together. And here's what she put together. Hospitals that had a lot of money had patients who wanted to know how their pregnancies were progressing. At that time, they would do X-rays to see how the pregnancies were progressing. They didn't have ultrasound then. So they would do an X-ray. So if you wanted to see how your baby was developing, you would go and get an X-ray picture. And as we all know, we all love to see our ultrasound. So what happened, people kept going back for more and more X-rays. And the more money you had, the more X-rays you could afford to have done. So what happened is she proved there was a link between birth defects and X-rays. People who couldn't afford to have an X-ray were having healthier kids. Rural communities, people who didn't have hospitals that were relying on midwives were having healthier kids. So she showed this link. And she was laughed at. She was, totally told that she was ridiculous. She presented her data. She stayed by it. She kept gathering data. And for 15 years, she was heckled and told that she was completely wrong. Finally, in the 70s, she finally got a couple of doctors that listened to what she was saying. And they, too, did a study with her. And suddenly it opened up the door and people realized that, X-rays were bad. So what happens with an X-ray? Well, when an X-ray hits our DNA, it can change the bases. When you are a fetus developing and you have a fewer number of cells, these types of mutations are much more damaging. You don't have 30 trillion cells yet, you're just little. And so every time you got hit with an X-ray, we would get different types of mutations that would occur. So she was the reason that we no longer have X-rays done today on pregnant people. Isn't that great? I'm so glad that she stood by her guns. So here's a picture of what can happen with X-rays or radiation. This is an example of UV radiation. A lot of times when we're exposed to UV or X-rays, we get the formation of what's called a dimer. A dimer is where two bases that are close to each other, when they get hit by a UV or an X-ray, they fuse and become like Siamese twins. They become like one. So they fuse together. So if I'm a segment of mRNA, when I read that as one base. So just like Siamese twins, when they're attached, they would be one person, they're attached to each other. That's how the mRNA is reading it. And so here, what's happened is we've had our little T's attached. We see this a lot when they're the same bases, like when we have two A's together or two C's together. What's going to happen is they'll attach together, and now they're going to get read as one base. This is going to be either an insertion or deletion example, right? Remember the one that we just talked about where I put a B in and everything was shifted? In this case, I've taken a base out and everything gets shifted, okay? So now when I come in here, when I'm trying to read this, I'm going to get the wrong message. Luckily, we have enzymes that are present that scan our DNA and look for these kind of effects, and they will snip it out and get rid of it. They're like little correcting enzymes. Isn't that awesome? Little correction enzymes. The problem that we run into, though, is if we get too many, it's like my Lego analogy. It's the difference between picking up one and a million. So if I have too many, I'm not going to be able to find them all and cut them all out. So as long as I'm getting exposed to UV light outside at a normal level, I'm fine because my enzymes can handle it. If I'm going to tanning beds or if I'm going outdoors long periods of time and getting burned over and over again, I'm running the risk of overwhelming my enzymes. Does that make a little sense to how that works? Okay. Yes. Thanks, Brianna. Yeah. So the idea then is that we have these dimer or these two bases. They form a dimer. I can fix them, but if I get overwhelmed, I can't fix all of them. Okay, I'm only a few enzymes. I can't fix them all. So again, UV light by itself, not that dangerous as long as you're just, exposed for short periods of time. But when you get exposed repeatedly, it's like an X-ray. It builds up. Just like how X-rays, you have to be careful to not repeatedly get exposed. We call those little enzymes that do these repair photolysases. Again, they follow the rule of ACE. And photolysase means that it's damage from the sun. So photolysases. Now, how do we know if other factors out there are mutagens? Well, it turns out that we can test to see if something is a mutagen, meaning it'll encourage a change in our DNA. And sometimes this can happen, and it can actually result in DNA changing to the point where it can become cancerous. Remember, cancer was uncontrolled mitosis. So maybe we affect the brake system in our cell that tells it to stop dividing. So that could be one thing we do. So the way that we can search to see if we have a carcinogen is to do something called the Ames test. The Ames test is where we're going to take a very special type of bacteria. It's a Salmonella type B. It's just an easy bacteria to grow. And what we're going to do is we have manipulated the genes on the Salmonella type B to not produce an amino acid that it needs to grow. It is missing the amino acid histidine, okay? So I have a bacterium. I have processed it, so it cannot make an essential amino acid that it needs for growth. All right, so I have Salmonella typhimurium. Okay. And what's going to happen is that I am going to come in here and I have removed an amino acid that it needs to grow. The one I remove is histidine. And I have stabilized this bacterium, so I'm growing it in my lab. And the way I keep it alive, because it needs this amino acid to live, is I grow it in broth with histidine. So it can get the histidine from its food. Okay, does that make sense? So I grow it in this broth with histidine. That's how I'm feeding it what it needs. Okay. What I'm going to do then is I'm going to take this bacteria and I'm going to put it into a situation where it's being exposed to whatever chemical I'm trying to test. let's say I want to see if gasoline is a mutagen. So I take a tube of gasoline, I put a sample of the Salmonella typhi in it, and I let it sit in there for a few hours. And then I'm going to plate the Salmonella typhi murium on a plate without the amino acid that it needs to live. Okay, on a plate without histidine. Now, if that gasoline caused my Salmonella to mutate, the reason we use this strain is one of the first things it will do with the mutation is it will recover the gene I took away. Everybody with me on that? So that if it is going to encourage a mutation, my Salmonella will be able to grow without me spoon feeding it that histidine amino acid. So I put it in media without histidine and I look for growth. Okay, so if I see growth, that means it's mutated. So I'm going to have some control plates set up. Now just make sure that my organism hasn't mutated while it was in the lab. Okay, so my control plates are here. They're the yellow ones. And then I have my test plate. This is the one that does not have histidine in it. Okay, there's no histidine. And if my organism has mutated, it will recover the gene and be able to grow on its own. All right. So then I look the next day and I see if I see growth. If I see growth, that means I have a mutagen, something that encourages change. If I don't see growth, then that means that I have a harmless substance. Everybody with me on how the test is working? What about over here? Look at my control plate. My control plate, I have 2 colonies. Uh-oh. Does that mean that I can't trust these results? Well, remember, I have a spontaneous natural mutation rate of 1 in a billion. So what that means is those are my 1 in a billion cells that did a natural mutation. All right? So this test is going to be something that can help us determine whether or not this is a product that we need to avoid. Okay, so here's an example of how other bacteria can also share or exchange genes. This is the process we refer to as genetic recombination. So we now know that DNA can change because of mutations, but here's another thing that's way to change DNA, and this is through sharing of genes, sharing genetic information. So this will happen when I have bacteria that contact each other and they are going to share plasmids. Remember we talked about plasmids before. What was a plasmid? Who can remember? Yes, from the general DNA. So yes, a plasmid is a small segment of DNA. And one of the things that it can carry is like antibiotic resistance, it can carry information, and our bacteria can exchange them back and forth. Kind of like Pokemon cards. You can trade them back and forth. You know, here's my plasmid, what's your plasmid, let's trade. So they do this all the time. I can have trades occur during reproduction between generations of cells, which means a mother cell could pass her plasmid on to her daughter. She'll be like, hey, this is really handy. When I replicate, I'll make a copy of my plasmid and I'll give it to you. So that's going to be a vertical transfer, giving it to its offspring. Or I can have it horizontal, which means I have cells that are the same age talking to each other and passing genes that way. Think of horizontal as like your friend, vertical as like your parent, okay? It just is which region you're getting the cells from, which type of cell you're getting things from. So with this, they're sharing information back and forth. And so that information, let's say that I pick it up from another cell, can get pulled in and I can keep certain segments and add them to my DNA and get rid of whatever I don't want. Now I can do this from interacting with another cell, or get this, they can do this just from DNA floating in the environment. So if I go in with an antibiotic and I kill bacteria, and those bacteria break open and fall apart, they're going to leave a little bit of their genomes behind because I popped them open, so some of their DNA is preserved. My immune system will clean that up. I don't have to worry about that. But while my immune system's trying to clean it up, these little segments can get picked up by neighboring cells. And then they can start incorporating these plasmids into their processing. Okay, so we can see that happen. So here's an example of how this process works. And this is actually an experiment that occurred when we first realized that genes could be shared between bacteria. Okay. So in this front picture, I have a picture of a mouse who unfortunately has died of pneumonia. Remember we talked about pneumonia. We said it's caused by Streptococcus pneumoniae. And it can have a capsule around it. Do you see the capsule that's around these cells? Remember, capsules increase virulence. They make it harder to treat. It's like an extra shield. Okay? So this mouse died of pneumonia, and the bacteria that it had contracted had capsules. All right. Now over here, we have a mouse that was injected with non-encapsulated bacteria. If I don't have a capsule, I don't have an extra shield. And so in this case, the mouse had a healthy immune system and was able to clear the bacteria on its own. Okay? Then what they do is they took this bacterium that was deadly, they heated it to see what would happen after heating it and injecting it into the mouse. And what they found was when they heated the bacterium with the capsule and injected it, the mouse remained healthy. Let's think about why. If I heat bacteria, does that kill the bacteria? Yeah, so I killed the bacteria, right? And all of the bacteria in there, all of them had capsules, they all died. When I injected it into the mouse didn't have a capsule, so it was just like what happened over here, and it survived. The difference between this mouse and this mouse is that I heat killed the bacteria. This is actually one of the ways we used to make vaccines in the early days. They would heat things to kill them off and then inject them. Okay, so that would build your immune system. That was the early days. But check out over here. This is a situation at the end where I've heated the bacteria. But then I took that heated broth and I put in non-encapsulated bacteria. So I put in the bacteria from my mouse here in B in with the dead bacteria. And guess what happened? They suddenly all started making capsules. How? Well, I had dead bacteria that had been heated, but their DNA was still present. Even though it's dead, just like when we die, we're still, we still have our corpses, we're still left behind. Okay. I put in bacteria without a capsule. They saw that DNA and they took that DNA inside of them and they went, oh, look at this. I found a recipe card on how to make a capsule. And they made a capsule and became as dangerous as that first column. This is why we are so worried when bacteria start doing mutations and becoming resistant. We worry because they can easily spread to other bacteria. And we just don't know. It's like shuffling cards. We don't know what we're going to get at the end. Any questions on this process? Okay. So here's another example of how bacteria can pick up genes. This is the one we mentioned before. Does anybody remember what this little bridge was called? It was called a sex. Yeah, excellent. Sex pili or sex pilis. Here's a little bridge that's being built between my cells, and they're shearing plasmids back and forth. Okay? So this is another way that they can exchange. The difference here is that these cells are both alive. So these cells are both living, they're actively sharing things back and forth. Again, if this cell suddenly died and it deteriorated and this little plasmid went into the environment, this guy could take it up that way too. Okay, that's what we saw with that previous slide. So here's another picture of a sex pilis in action. So we call that bacterial conjugation, and they're exchanging genetic material back and forth. We, as I said before, we really don't know a lot about this process. All we know is that it occurs, they exchange genes, but we still don't know how they signal one each other. How do they know who has which Pokemon cards? How do they talk to each other? We don't speak the language of bacteria yet. We wish we did. But we know that they send each other signals. What's fascinating is they can be even different bacteria. I can have E. coli talking to Salmonella. So they speak the same language even though they're different bacteria. That's trippy, isn't it? I mean, think about that. They're from totally different backgrounds and they're speaking the same language. This is cool stuff. There's a whole secret world that we haven't even touched on yet. Yeah, Aaron.
So is it the one that contains the plasmid that initiates the
Yes, that at least we think. We think that's what it is. We think the one with the plasmid must put out a protein of some sort that says, hey, I have something cool. Kind of like putting up an open sign, like, hey, come see me, I have something cool, you know? We think that's what it is, more so than the other one who doesn't have it kind of bumping into a whole bunch of cells going, do you have anything? Do you have anything? We think it's more like they send out a chemical attractant saying, come see me. They're almost like a little dealer, you know? They're like a little dealer, like, here, come here, kid, come here, you know? Sometimes it goes bad, you know, it goes really bad. Just like sometimes drugs go bad, you know, well, usually drugs go bad. It's like, you know, things go bad. So, E. coli 0157. Remember I mentioned E. coli lives in our gut and it's harmless? E. coli 0157 is one who unfortunately hung out with a drug dealer. He ended up hanging out with a Shigella that gave him a really nasty toxin and forever changed him. Okay, so he like went on the dark side, all right? So sometimes things can go really wrong when this happens. Okay, let's talk a little bit more about how mutations can occur then. So as we've mentioned, mutations can occur from outside sources. They can occur naturally during replication. They can occur from exchanging genes back and forth. Now, when this happens, we create a new bacterium. And the reason the bacteria are constantly exchanging genes or wanting to mutate is because they're always wanting to figure out a better survival strategy. If we're hitting them with antibiotics, of course, they're going to want to build a resistance to that. They're always trying to get ahead of us. Okay, so we're in this constant race with them. And so their environment is going to push them to keep becoming different, to keep becoming better. So natural selection, this is kind of what is behind the idea of evolution. Natural selection says, our environment says what's going to survive. So we're seeing that, like I said, with COVID. We have seen so much death. But we've also seen a lot of people who are naturally immune. Why? Because they carried a genetic combination that worked with this virus. They was able to fight it off. And so if we hadn't had a vaccine come out, if we hadn't had intervention, we still would have naturally had some people who were just immune because of mutations. So mutations are not always bad. There's usually an environmental factor that's going to push. With the Black Plague, we mentioned before that it wiped out a huge percentage of Europe. In some areas, it was like a third. In some areas, it was 2/3, which is phenomenal. I can't even picture that. A huge percentage of Europeans. If you test Europeans nowadays, the ones who are still alive still carry a slight resistance to the Black Plague. That was the impact it had. So people who lived had a resistant gene, and they still have it to this day, which is fascinating that we can find that. It's pretty interesting. Now, this brought up an interesting question then. Why, if the environment's dictating these changes, why would this possibly, be helpful, and how can we use this when we're studying diseases? And we started looking closer at genetic diseases. So in other words, why are genetic diseases present? If they're so, debilitating, why is this getting passed along from one generation to the next? And we found some interesting links. I've told you bacteria have been messing with us since the beginning of time. Well, here's a great example. Okay, sickle cell anemia. Anybody heard of sickle cell anemia? I see lots of nods. Good. Sickle cell anemia is where instead of having a nice round donut-shaped red blood cell, I get a sickle crescent-shaped red blood cell. Now, depending on what kind of genes I'm carrying, I can either be a homo and heterozygous. So I can either have like a normal gene, we'll say a capital N for normal, so I can have two normal genes, or I can have a normal with the sickle cell trait, or I can have two sickles. This is the one I worry about. If I have two recessive sickle cells, then that means that I'm going to have sickle cell anemia. Okay? So the question becomes, why did this evolve? The concern, if I have sickle cell anemia, my cells cannot carry oxygen like normal. They actually get stuck and form clots a lot of times. A lot of people who have sickle cell anemia can't live past the age of 40 without constant blood transfusions. We usually see that they have to have hip replacements at around the age of 50. because of blood clotting that starts to kill or break down the cartilage in the joints. So we start to see hips going bad, shoulders going bad. So this is not an easy life to live. So why is it still around? Well, it turns out that, as I said, it's a recessive gene. And so if a child has two of those quote unquote bad genes, the two S genes, then they are going to have the trait of sickle cell. If they have an A and an S, the A is standing for normal in this case. If they have an A and an S, what that means is they're going to have half normal cells, half sickle cells. So they're carrying it, but it's not enough to cause all this damage and shorter life expectancy. Okay, so why would this evolve again? Well, here's a fascinating picture. Here's a picture showing where sickle cell originated and is primarily found. And here's a picture showing where malaria originated and is primarily found. And it's been slowly spreading across the land. So as you can see, initially malaria here and sickle cell here matched up very well. So the question then became, okay, why? Why are we seeing this develop where malaria is? Well, what we found is if I have normal, healthy red blood cells, I tended to have a higher death rate if I was exposed to malaria. The reason for this is malaria is an organism that is microscopic. It's carried by mosquitoes. It will get into our bloodstream through a mosquito bite. It will infect our red blood cells. And what it does is through its life cycle, it will explode or burst the red blood cells. If I have all these nice normal red blood cells, that means I'm going to get a very high load of malaria. And when all of my cells burst, I can go into shock and I can die. Not a good outcome. But check out my carriers. People who are carrying the trait, half sickle, half normal. It turns out malaria cannot infect sickle cells. It goes up to them, but it can't attach. So if I get a malarial infection, I have only half the infections because I don't have as many red blood cells that can be infected. And when those red blood cells burst, I have the sickle cells that are still able to function. They're not great, but they're limping along. You know, it's like my old car that I'll drive to work. It's not fantastic, it's not flashy, it's not fast, but it gets me there. Okay, same idea. So it's getting us there. It's enough to get us through a malaria outbreak. And then we survive and we move on. If, unfortunately, I get the sickle cell trait, the one at the end, then I have a chance of early death. So what does this tell us? This tells us that genetic defects actually were survival mechanisms. If we were a carrier for some of our genetic defects, it would protect us against some of the bacteria, viruses, plasmodiums, different organisms that are out there. Isn't this cool? This is cool stuff. So another one that we're studying right now, cystic fibrosis. Cystic fibrosis is a genetic defect. It affects enzymes in the body, it affects salt concentrations, all these things. What they found, though, is people who are carriers for cystic fibrosis are naturally showing high immunity to cholera. So cholera used to be something that contaminated water. And so it's a survival mechanism. And so we're revisiting all of the genetic diseases now and trying to figure out things from an old point of view. We have all these medical interventions nowadays that we do to keep people alive, but we're putting ourselves back in the past and saying, how would this help us when there wasn't anything medical around? And we're finding a lot of correlation. Bacteria have been messing with us since we first walked on this planet. Okay, I'll go ahead and stop here. So it's our last slide. Hallelujah, right? And then what's going to happen is that we are going to take our break. Do you guys want an extra, like 15 minutes to study? Do you want to start lab at 10:45? Okay, we'll start lab at 10:45. So you can stay in here if you want. You can move somewhere else if you want. Totally up to you.